So to start off, thank you to the organizers for selecting this abstract for a short talk, and thank you all for tuning in today. I am a PhD student in the Plakino lab at the University of Cambridge, and as mentioned today, I'm going to talk about the time-resolved single-cell RNA sequencing profiling of myeloid cells after spinal cord injury, which has identified a novel Fabp5 expressing subpopulation of inflammatory myeloid cells in chronic spinal cord injury. The data that I will show today can be reviewed on our preprint and at the poster session later today. So to get started, traumatic spinal cord injury is a highly debilitating pathology that afflicts thousands annually, irreversibly reducing quality of life, and currently no fully restorative therapies exist. The pathology is highly dynamic, and it begins with an initial mechanical insult that compromises the structural integrity of the spinal cord and the blood-spinal cord barrier, and initiates a cascade of biological events known as the secondary injury, through which the damage continues to spread to the adjacent tissue. The secondary injury is made up of three main phases: the acute, subacute, and chronic, and each of these are highly dynamic and multifaceted, and I don't have time to go into all of them today, but we'll focus on a common component that's present throughout the course of the pathology, which is inflammation. So unlike in normal wound healing, where inflammation resolves within the first few days, in spinal cord injury, we have persistent inflammation, which extends indefinitely, disrupting transition to later phases of wound healing, resulting in a chronic wound state of impaired recovery. And interestingly, in normal wound healing, myeloid cells return to normal levels within weeks. But here, just like in other chronic wound states, after spinal cord injury, myeloid cells remain elevated. So what are myeloid cells? Well, this is an umbrella term for a diverse set of immune cells that respond to tissue damage and pathogens. So in a central nervous system parenchyma, we have microglia, which under healthy conditions maintain their populations in the brain through local proliferation and have a lifespan of over 1 year in mice and even longer in humans. And in contrast, the peripheral myeloid cells, such as macrophages, arise from hematopoietic stem cells in the bone marrow. And under healthy conditions, they will circulate in the blood and are very short-lived, with a typical lifespan of up to 3 days. But upon spinal cord injury, infiltrating macrophages can take on the morphology and transcriptional profiles of microglia, making them very difficult to tell apart. And it is clear that both types of myeloid cells, so both these infiltrating macrophages and the microglia, drive inflammation after spinal cord injury, but the transcriptional pathways behind these responses is not yet understood. So this brings me to our hypothesis, which is that a specific myeloid cell identity disrupts the progression of normal wound healing and could be targeted to resolve the chronic wound state. But how can we overcome this challenge of disentangling microglia from infiltrating macrophages? Well, in this study, we used a fate mapping mouse line, which we dubbed the cremato-mouse, and it takes advantage of the differing lifespans between microglia and infiltrating macrophages. So with this mouse line, tamoxifen administration and then a 28-day washout period leaves microglia as double positive for YFP and RFP, while the infiltrating myeloid cells, including the macrophages, are YFP positive only. And this tool allows us to isolate these myeloid cells and study their respective roles after spinal cord injury. So to fully address our hypothesis, we used this cremato-mouse line to generate a mouse model of moderate contusion spinal cord injury, and we collected the tissue from the lesion epicenter or sham-injured controls at 5 different stages after spinal cord injury, ranging from the acute to the early chronic. Thanks to the cremato-mouse, we then used FACS sorting, or FACS, to isolate microglia and infiltrating myeloid cells, and then we performed single-cell RNA sequencing using the 10x Chromium platform. After quality control, our data set comprised nearly 31,000 myeloid cells from the lesion epicenter across 5 time points and the sham-injured controls. So if we investigated the expression of cell type markers in the dataset by averaging the log10 UMI counts of the listed genes and then visualizing them on a UMAP, you can easily see that we were able to identify all of the expected myeloid cell types. And thanks to the chromatomouse, we were able to identify a population of infiltrating macrophages that have taken on microglia-like phenotypes. And additionally, we can also be sure that despite the downregulation of these classic microglia markers after spinal cord injury, that these cells here are in fact microglia. So now I'd like to direct you back to our hypothesis. And in order to address this, we first clustered the cells, then we characterized them using trajectory analysis, differential gene expression, and gene ontology analysis. So in the interest of time, I'll shift the focus towards the microglia data, but if anyone's interested in the dynamics of the other myeloid cells, please check out the preprint. So we performed trajectory analysis using the Monocle 3 package. And since the experimental design encompassed several time points, we were able to give directionality to the trajectory. So microglia after spinal cord injury can be split into two trajectories, an A trajectory this way or a B trajectory. The microglia B trajectory is very clearly cycling, which is in line with previous findings that found microglia to proliferate for up to 7 days after spinal cord injury. The microglia A trajectory, on the other hand, is more complex, and so we'll go through each of these clusters together. So cluster A1 is dominated by cells from day 1 post-injury. Based on the GO analysis, it appears to be pro-inflammatory and involved in lipid transport and the response to oxidative stress. And notably, this cluster is marked by the expression of fatty acid binding protein 5, or Fabp5, which is an intracellular lipid transporter involved in fatty acid uptake, transport, and metabolism, and has pro-inflammatory effects in other contexts. Cluster A1 also downregulates CSF1R, which is a gene required for microglia proliferation and survival, and yet this cluster shows no signs of apoptosis, at least from the transcriptional level, and in fact, according to the trajectory analysis, they seem to project into the subacute and chronic phase cluster microglia A2. So A2, cluster A2, is dominated by cells from day 3 onwards. It appears to be cytotoxic, phagocytic, lipid processing, and has a continued pro-inflammatory response, which would negatively regulate canonical wound healing functions. Cluster A2 seems to be split by the mutually exclusive expression of CSF1R and Fabp5. And cluster A2 cells collected at later time points show increased Fabp5 levels. So it could be the case that the downregulation of Fabp5 allows cluster A2 to transition into cluster A3, and without this downregulation, cells would remain in a state of cytotoxicity. It's also worth briefly mentioning that thanks to our chromatomouse, we were able to see that cluster A2 is also contributed to by infiltrating macrophages, which seem to have adopted microglia-like profiles in the subacute and chronic phases. And now finally, we have cluster A3. So this cluster is dominated by cells from day 10 onwards. It's characterized by a shift towards homeostasis and anti-inflammatory responses. And interestingly, this cluster upregulates many of the neuroprotective microglia genes that were recently described in mouse models of Alzheimer's disease and ALS. And the cells that express Fabp5, regardless of their cluster, express lower levels of these neuroprotective genes. So if Fabp5 really does prevent cluster A2 from transitioning to A3, then as we hypothesized, Fabp5 positive A2 cells could disrupt the progression of normal wound healing and could be targeted to resolve the chronic wound state. So what I've shown you so far is all single-cell RNA sequencing data, so of course it needs further investigation through complementary methods in order to verify our findings. And we are currently working on this. One of our approaches is to use single molecule fluorescent in situ hybridization, or single molecule FISH. So here you're looking at some SM FISH data at the lesion epicenter, and you can see that, as in our single cell RNA sequencing data, Fabp5 expressing myeloid microglia are absent in the sham-injured control. They are present in the acute phase, possibly as our microglia A1 cluster, and they also persist until day 21, again likely as our microglia A2. I'd also like to draw your attention to the morphology of these microglia. So in the control, you can see that the microglia have this nice ramified morphology, but by day 3, they are highly activated and amoeboid. And then by day 21, the Fabp5 positive microglia, denoted by the white arrows, remain amoeboid, while the Fabp5 negative microglia, with this red arrow here, seems to have recovered something of this ramified morphology. So to summarize, we believe that the Fabp5 positive cluster A2 cells, which are contributed to by both microglia and a population of subacute and chronic infiltrating macrophages, may disrupt the progression of normal wound healing after spinal cord injury. And as I mentioned, this all requires further investigations. Some of the things that we're working on are providing some spatial context using immunohistochemistry and SM FISH. Other things that we're working on are functional studies, which are going to be really important to understand the actual role here of Fabp5. So we're working on an in vitro study right now to knock down Fabp5 in myeloid cells. And we also have a Fabp5 specific knockout mouse line under a myeloid lineage promoter. So we're going to investigate the effects of myeloid cell Fabp5 knockout after spinal cord injury. And with that, I would like to thank my labs, the Bucchino Lab, the Marioni Group, and our collaborators, as well as all of our funders. And I'm very happy to take any questions.
Thanks, Regan. That was awesome. So just to jump into questions here, Michael says, very nice data, and asks if you could comment on how cell death pathways are perturbed along pseudotime in your injury model.
Sorry, how cellular pathways are?
How cell death pathways are perturbed along pseudotime in your injury model.
Cell death pathways? Sorry, I can't quite hear you.
Cell death.
Yeah, cell death pathways. So according to the literature, there should be a substantial amount of apoptosis after spinal cord injury. And we are really not seeing much of that in the data, but I think that it's because those cells aren't passing our quality control. So if we have much looser QC metrics, we'll probably get some more of those dead and dying cells, but we are looking more towards the ex vivo data to try to characterize some of that apoptosis and necrosis.
Very cool.
Homera asked if you could elaborate on what other processes are going on along the different time points, other than inflammation, remyelination, regeneration, etc.
Yeah, so of course it really depends on the cluster and the time point. But if we start with the microglia A1 cluster, which temporally is occurring first, of course there's lots of inflammation, but there is also the start of some phagocytosis. We have some lipid processing as well, and that seems to be carried over until the microglia A2, where we have no signs of regeneration yet. So again, as I mentioned, that's a lot of inflammation in this cluster A2, but then a lot of cytotoxicity, a lot of ROS production, which kind of goes hand in hand. But then once we get into that cluster A3, the one that seems to be contributed mostly by subacute and early chronic cells, that's when we start to get these go terms for regeneration or axon regeneration. Of course, we're looking at myeloid cells, so you can't really say that that's coming directly from them, but hopefully they're having some kind of slightly regenerative effect. But if you look at the pathology in mice, at least, you don't see a ton of regeneration. You see a little bit, but obviously these animals do not become fully repaired like a salamander or something. Yeah.
So Regan, Chris McGinnis asks if you could comment on the role of lipid metabolism on macrophage polarization in this context, and really is asking, is the role of Fabp5 mostly metabolic?
It's a really, really good question. We are investigating that in our in vitro studies. We're really, really interested to see what Fabp5 is doing when it comes to the polarization between pro and anti-inflammatory. So that's something that we're doing in vitro. We're trying to challenge these myeloid cells with canonical M1 or M2 stimuli, and then also having Fabp5 knocked out. And then we are trying to measure different metabolic outputs, but also different cytotoxicity outputs. So yeah, it's definitely unclear at the moment, but we're hoping to get a better picture soon.
Okay, great. Let's take one last question from Elizabeth Fisher. Nice talk. Can you distinguish between border-associated macrophages from infiltrating macrophages in your data?
Yeah, so border-associated macrophages are typically long-lived, like the microglia. So with our chromato mouse, we would expect them to be double positive as well. But despite the similar labeling that they would have through the fate mapping mouse line, they do have distinct transcriptional profiles. And I've looked extensively through the data set for them, but have not found any substantial population of border-associated macrophages in the data set. And I've looked at other myeloid cell data sets using this exact same mouse line, and we have identified the border-associated macrophages in those tissue samples, or single-cell data from those tissue samples, but they came from whole brain and spinal cord homogenates, whereas the data set that I was showing you today is just from a very small 5 millimeter section centered on the lesion. So I'm thinking that they're probably quite a rare population compared to all of the other myeloid cells that we have at the lesion epicenter, and that's why we're not really detecting them.
Awesome. Thanks, Regan, for a fantastic talk once again.